She will give us a presentation on the first isolation of an ancestral herpes virus from Great Lakes lake sturgeon. With that, I will give the phone to Amber.
All right, thank you so much for the introduction. So I'd like to start out by acknowledging my co-authors for their contribution on this research that I'm extremely excited to share with you today. And those collaborators come from MSU, Michigan DNR, and the University of California, Davis. So to kick things off, lake sturgeon are the only native sturgeon species in the Great Lakes, where they're an ecologically, culturally, and historically important fish. Since the 1800s, when they became a valuable commercial fish, their numbers began dramatic declines. Now, lake sturgeon populations are estimated to be at less than 1% of historical numbers. And because of this, substantial resources have been allocated to the hatchery rearing of lake sturgeon for conservation purposes. Hatchery rearing of lake sturgeon is now a common practice around the basin. These facilities, some pictured here, are populated exclusively from gametes collected from wild adults. And unfortunately, these facilities are not exempt to troubles of their own. State, federal, or tribally managed facilities all grapple with occasional mortality events, sometimes nearing 100% mortality. And one of the unique things about lake sturgeon is just how prehistoric they are. And from what we know of other sturgeon species, many of the pathogens that affect them are quite distinct from other fish. And many cannot be detected without specialized and specific diagnostic tests, which is likely the factor behind our continued inability to figure out what's causing these mortality events. So while this is happening, other sturgeon pathogenic microbes seem to be increasing in host and geographic range. And despite an apparent encroachment towards the Great Lakes, the potential impact of these sturgeon pathogenic microbes, should they end up in the basin, is largely unknown. And unfortunately, because these pathogens aren't detected with routinely employed diagnostic techniques, we know very little about their potential presence in the Great Lakes. And that's where my thesis research is hoping to uncover some unknowns. If you attended Symposium last year, you may have seen my talk that focused on the overall scope of my ongoing thesis research that looks to assess the risk of emergent and endemic fish pathogens to Great Lakes lake sturgeon populations. For the next few slides, I'm going to bring you up to speed on what's been done as a part of this larger project before diving into my most recent findings. In the first year of this larger study, I sampled adult lake sturgeon from the Peshtigo River in the Lake Michigan Basin, the St. Clair River in the Lake Erie Basin, and the Black River in the Lake Huron Basin. During year one, I non-lethally sampled an array of tissues and fluids from over 230 adult lake sturgeon. We collected these samples with the intent to employ specialized diagnostic methods to detect a wide array of pathogens, including sturgeon pathogenic microbes. And during year one, we observed these circular, raised, semi-translucent areas of apparent cutaneous thickening. These lesions varied in location, and they also varied in intensity, from focal to more severe, sometimes coating surfaces of the fish. And in terms of how frequently we saw these lesions, they were observed at a prevalence of 5.8% in the Black River in the Huron Basin, and at a prevalence of 1.3% in the St. Clair River in the Erie Basin. So upon doing some digging into what could possibly be causing these lesions, I came across multiple references referring to acid herpes virus 1 and 2, both in the family Alloherpesviridae. And based on the literature, the lesions I had collected from adult lake sturgeon appeared grossly similar to those noted in other affected sturgeon species. And for that reason, I began pursuing specialized diagnostic techniques centered around the detection of acid herpes viruses. Armed with that information, I optimized a PCR assay capable of detecting a wide array of fish viruses, including those in the family Alloherpesviridae. I screened the lesions using this PCR and successfully detected an amplicon from an unknown virus associated with these lesions. I then sequenced the amplicons and analyzed them phylogenetically, and what I'm showing here is the resulting dendrogram. Highlighted on the right are my sequences, and from this, we determined we definitely found an alloherpes virus, and it was most similar to acid herpes virus 1, yet distinct. So while I found genetic traces of a virus, unfortunately, and despite my best efforts, I was unable to isolate the virus in vitro, which is critically important for future studies. Though disappointing, this wasn't exceptionally surprising, given that viruses within the family Alloherpesviridae are notorious for being difficult to isolate via cell culture. So why exactly was this finding so significant? Well, acid-censored herpes virus 1 and 2, also known as white sturgeon herpes viruses, are listed as emergency pathogens on the GLFC Fish Health Committee model program. They earned their spot on this list due to their ability to cause mortality in other sturgeon species and be transmitted from adults to offspring. Nobody thought an acid-censored herpes virus was here. We certainly didn't want it to get here, and we may have just found it. So while my previous research began to answer some critical questions, it also raised many, many more, just a few of which are, what exactly is this virus? We couldn't isolate it, how can we? Is it contributing to population declines? And could it be behind these undetermined mortality events in hatcheries? And that brings me to my overarching study hypothesis, which is that a Great Lakes acid-censored herpes virus is negatively affecting the health and survival of Great Lakes lake sturgeon. To answer our hypothesis, we developed 3 key objectives. The first is centered around elucidating the effects this acid-censored herpes virus could be having on wild adult lake sturgeon. So to begin understanding these effects, we went back to tissues collected from lesions. I'd seen gross changes in the skin caused by these lesions, but also wanted to know what was happening microscopically. But before we get to what I found, I'm showing you a skin section collected from a grossly normal area, in which you can see an epidermal layer of normal thickness and the underlying dermis. Now what I'm showing you is tissue collected directly from a plaque-like lesion. We immediately noticed how thickened the epidermis was, and we also noted inflammation within the epidermis and underlying dermis. Additionally, we found other evidence of tissue damage in the form of swollen degenerate epithelial cells noted here with red arrows. And we also found evidence of intracellular edema, or fluid accumulation between the epithelial cells. Collectively, what this told us was that in lesions where we found the virus molecularly, we had evidence that there were negative changes in the skin. And this is particularly important given the role the skin plays in pathogen defense and host osmoregulation. So because of this, we concluded that the virus could possibly be negatively affecting wild adult lake sturgeon. Our second objective was to isolate the virus in vitro. While the detection of genetic material, again, was promising, one of the pillars for linking a virus to disease causation is fulfilling something called Rivers postulates, which requires infectious virus experiments under controlled laboratory conditions. And to do that, we needed to be able to grow the virus. So now that I knew I was dealing with an alloherpes virus and had been able to research different methods to effectively isolate it, I was super excited to get into the field in 2020. Though the ongoing COVID-19 pandemic certainly posed challenges. Despite that, though, their perseverance and collaboration, I was able to get out to the Black River and non-lethally sample lake sturgeon. And let me tell you, hiking a river, all the while decked out in the proper PPE, certainly wasn't the most enjoyable experience, but one I'm very thankful for and will remember nonetheless. So moving forward to what I saw while I was in the field, I saw multiple fish exhibiting these acid-censored herpes virus linked lesions in varying intensities, similar to the year prior. These lesions were observed at a prevalence of about 14%. So now that I had these lesions and had done research to develop new sample handling techniques, I was able to employ my new viral isolation methods. And based on my research, I was very optimistic. So after implementing my modified sample handling protocols, I inoculated lesion tissue onto cells. Soon after, I saw evidence of a replicating agent in the form of multinucleated giant cells, noted here with blue arrows. Eventually, that became even more widespread, as you can see here, again noted with the blue arrows. And this finding was very encouraging to me because last year I didn't get this far, and this year I did it. So since we saw evidence of a replicating agent on cells, we wanted to look at them via transmission electron microscopy. And when looking closer, we appreciated enveloped icosahedral-shaped virions, which appear as hexagons in 2D here. And they also resemble other alloherpes viruses. Excitingly, when we looked at the tissues, we saw the same shaped virions, and though in this photo, it's much easier to appreciate the surrounding envelope. This suggested that the replicating agent in the cells was likely the same virus as in the skin lesions, at least ultrastructurally. Additionally, we assayed affected cells using the same PCR assay as the year prior, and once again got similar positive results. So when using a PCR assay that amplifies multiple targets, there's always a risk of non-specificity. But cloning lets us take that into account and be more confident with our sequence results. For those unfamiliar with cloning, very briefly, the cloning process starts with an unpurified PCR product that's then purified, inserted into a cloning vector, plated on selective media, and the resulting growth will consist of clones of the PCR product inserted into that vector plasmid. And this all lets me be more certain that what I generated with my PCR is my target and not something else. So what we noticed is that throughout the tissue samples, tissue culture samples, and clone samples, the results we generated were highly similar, further supporting that what we had found in the tissues was the same as what we found in the cells. And lastly, since we were able to isolate the virus in vitro, I wanted to elucidate the effects this virus has on juveniles. Does it induce disease and/or mortality? That's a crucial knowledge gap that I needed to address. So this year, I collaborated with the Black River facility crew to rear juvenile lake sturgeon and held them under quarantine conditions at the University Research Containment Facility here on campus. So now that we also isolated this virus and could readily grow it, the possibilities, quite frankly, felt endless. And with so much unknown, we began several pilot challenges to evaluate the effects of dose, temperature, and exposure. To do so, I grew this virus up in the lab and either immersed or injected the fish. After exposure to the virus, I monitored them for the development of disease signs. Over time, I began to note a variety of things. First was perioral hemorrhaging, as seen here and noted with the blue arrow. Second was varying degrees of exophthalmia, or swelling of the eye, which both of these are noteworthy because some of these disease signs have also been reported in sturgeons affected with other acid-penciled herpes viruses. We also observed ulcerative skin lesions in the infected fish, and they varied in intensity. Some were more focal, some were much larger, and in other cases, these ulcers were so severe that entire fins had been eroded away, as you can see here. So now, based on these pilot studies, I saw that the virus could induce disease, but what effects did it have on survival? In all pilot experiments, I found that the virus could induce mortality. And what I'm showing you here is the mortality induced in one of these pilot challenges, with the days post-infection on the x-axis and cumulative mortality on the y. So what you can see is that no mortalities were observed for the first 52 days of this study. But once they were, the cumulative mortality leaped from 0 to 60% in just 5 days, in addition to another mortality just 3 weeks later. And this was pretty interesting because while it definitely took some time, it seemed like the disease course of this virus was pretty consistent. So the results of these pilots were very telling and concluded that the virus can induce mortality in juvenile lake sturgeon. Additionally, this also helped me structure full experimental challenges. In fact, my injection challenge is currently ongoing, and within the next few weeks, I'll also be starting an immersion challenge. So to wrap things up, we have a few key conclusions. First is the report of an acid-penciled herpes virus in Great Lakes lake sturgeon. As I said before, there are currently no published reports of an acid-penciled herpes virus in the Great Lakes. And given the devastation viruses in this family have caused in other sturgeon populations, as well as their status as an emergency pathogen in the GLFC Fish Health Model Program, this finding was significant. Second is the very first in vitro isolation of an acid-penciled herpes virus in the Great Lakes. And while initial attempts at isolation were unsuccessful, using sturgeon-specific cell lines and modified protocols, our research crew is now able to successfully isolate, cryopreserve, and revive active virus for further studies. And third, through that work, I've begun to elucidate the effects this virus has in adults and juveniles. And unfortunately for lake sturgeon conservation, I found that this virus can kill juveniles and is clearly present in spawning adults. Has this virus been working in the background, contributing to these declines in lake sturgeon? Or has it been a factor in the unexplained mortality events happening in breeding facilities? My research is positioning us to answer those critical questions. So with that, I'd like to acknowledge the U.S. Fish and Wildlife Service's Great Lakes Fish and Wildlife Restoration Act for funding my study, the Black River Sturgeon Facility crew, Michigan and Wisconsin DNR, MSU Aquatic Animal Health Lab members, and my major advisor, Dr. Thomas Locke. And I will take any questions.
Thank you, Amber, very much to give us this interesting talk. I saw a question from Gary.
Yeah, thanks. Amber, nice talk. I'm not surprised at the slow mortality. Herpes viruses are notorious for slow replication. So it obviously hit the necessary point to cause mortality. I am curious in some of those fish that were showing clinical signs in the caudal peduncle that basically lost their tail fin for all intents and purposes. Were you able to extract any virus from that wound?
Yeah, so all 3 of the lesions I showed photos of, I did successfully re-isolate virus from.
Thanks.
Any other questions? Oh, I see a question from Jean.
Yeah, I was wondering if you had some control batches of fish that didn't die in the juvenile studies.
Yep, so for all pilot studies that I've done and also my full challenges, they're all done with a control group and a challenge group. So equal numbers of fish in each group and equal numbers of replicates in each group.
Thanks.
Yep.
We got another question from Jean. Jean Chao.
Hi, Amber. It was a very nice talk. Sorry I missed the very beginning, so I hope my question, won't be due to that. But I was just curious because I did see the picture, you and others in the stream in the river. I was curious if you have seen any difference in prevalence of infection with age of the sturgeon or severity or anything like that.
So right now we haven't looked at anything to do with age. We've only really looked at the presence of lesions and the severity of the lesions when we find them. So there hasn't been a correlation made with age.
You mean it's not been obvious, or you haven't looked quantitatively?
We haven't looked quantitatively.
Okay. But no gut feeling. You're not willing to go out on a limb.
I wouldn't say there's a gut feeling.
Okay. All right. Thanks.
Yep.
Are there any questions from our audience for Amber? Yeah. If not, thanks, Amber, for the great presentations. And